Hey Team Bio, welcome to your screencast on protein structure. So we have talked about the model for enzyme action, the induced fit model, where we saw a enzyme binds a substrate, then in the active site it changes shape, and then it catalyzes the reaction by forcing the reaction to take place by putting the substrate in a really specific position. Then it ejects its product, and then it changes back into its original shape again, so it's ready to go again. And this screencast is all about how proteins make their shapes. So it's about protein structure, but I guess it's also about the interplay between structure and function, because as you know, a protein structure is integral to its function. If the structure changes, then the function is going to be changed as well. Okay, so we're going to be talking about the 3D shape of a protein. Specifically, we're going to be talking about enzymes and their three-dimensional shapes and the levels of organization that let the shape take place. Okay, so up here in the upper right-hand corner, we have two amino acids. They're monomers. So just single amino acids here. And when they undergo a dehydration synthesis reaction, they form what we call a di-2-peptide. And this is actually a reaction that you did in class today, or depending on when you're watching the screencast, a day or two ago. Okay, so when two amino acids come together with a dehydration synthesis reaction, they form a dipeptide, and obviously, I forgot to mention, a water molecule. That's part of dehydration synthesis. And when this happens many times over, so if another amino acid were to join on to this end of the chain or this end of the chain, we would create a polypeptide. So that's what this is showing us here. So add more amino acids, and you get a polypeptide. So each of these is an individual amino acids. Maybe this is methionine, and this is phenylalanine, and this is alanine, and this is valine, and so on and so forth. There are 20 different types of amino acids. These are all these black lines are representing these peptide bonds. A peptide bond is, as we said up here, it's formed by dehydration synthesis, and it's a form of covalent bond. So that means it's very strong. Of all the bonds that we've learned about, ionic, hydrogen, and covalent is the strongest. So all these black lines represent covalent bonds, and this long chain of amino acids is our polypeptide here. So this is what represents our primary structure. It's the sequence of amino acids, methionine, phenylalanine, alanine, valine, and so on and so forth. If we were to change the sequence, so say we substituted out an alanine here for a lysine, then we've changed the primary sequence. The primary structure is the sequence of amino acids. Okay, great. Secondary structure is right here. So now when we take our primary structure protein, our polypeptide, which at this point is not folded in any way. It's just a long, straight chain of amino acids. If we take that polypeptide and we fold it into a specific shape, we're going to get our secondary structure. So it's the first level of folding. There are two folding patterns that I want you to know. I want you to know this type of folding pattern. It's called an alpha helix folding pattern. It's kind of like a spiral staircase folding pattern. Now, I don't know if this is helpful, but I took my polypeptide and I tried to turn it into an alpha helix from up here. It's not very good. This is the alpha helix shown from your textbook. They do a better job of it, I think. So they're showing you here amino acids folded up together to form this alpha helix. These dotted lines here represent hydrogen bonds. So the primary structure, what's linking the amino acids together one to another, so this amino acid to this amino acid, it's a little harder to see individual amino acids in this chain, are the covalent bonds, these peptide bonds. They're really strong. But what's holding the secondary structure together, this alpha helix staircase type of structure, are hydrogen bonds. And I guess I could draw some hydrogen bonds in here. So there are going to be hydrogen bonds inside of this alpha helix holding it together. So I guess that what I'm trying to say here is that while the primary sequence, this sequence of amino acids, because it's held together by covalent bonds, it's very strong. It's really hard to break up a polypeptide. It's not that hard to unravel an alpha helix because you just have to break hydrogen bonds, which are much weaker. Okay, the second type of secondary structure, which I want you to know, is called a beta sheet. It's also sometimes called a beta pleated sheet, but we're just going to call it a beta sheet. Again, here's my drawing of a beta sheet. And much like the alpha helix, there are hydrogen bonds in between the layers of the sheet holding it together. Here's your textbook version of the beta sheet. It's probably a lot easier to see. I think what your textbook does a bad job of doing is showing that these actually loop around and they're all part of the same polypeptide. My drawing does a very good job of doing that. So you can see here, it's all one long chain folded into this beta sheet. But also, I want you to take away here that the same thing is true of the beta sheet as the alpha helix. The structure is held together by these relatively weak bonds, these hydrogen bonds. So it's hard to break apart the primary structure, but it's not that hard to break apart the secondary structure. Okay, the third level of folding is called the tertiary structure. And the tertiary structure is, it's just when you get a long enough chain that you have two or more folding patterns in one polypeptide. So here is the image from your book, and they've highlighted two areas of interest within the protein. You can see this area, this is an alpha helix, and this area is a beta sheet. I've tried to do the same thing with my drawing. Here's my area of alpha helix, and here's my area of beta sheet. Okay, tertiary structure is held together by a couple different types of bonds. So again, hydrogen bonds play a role. They're definitely responsible for the beta sheets and the alpha helices within the tertiary structure. And here's another beta sheet you can see right here. They also might be responsible for like this kind of curlicue that you see right here. So hydrogen bonds are important. Other important groups are hydrophobic R groups. Within the amino acids, they tend to clump together because they don't want to interact with polar things. And then hydrophilic groups hydrophilic R groups of amino acids also tend to cluster together. Because proteins are all occurring in aqueous solutions, that means water-based solutions, these hydrophobic R groups tend to cluster on the inside of the protein away from the water outside, and these tend to cluster outside, close to water. Okay, the last type of bond that holds this all together, these are called disulfide bridges. They're bonds between two sulfur molecules. Okay, these disulfide bridges, they're covalent bonds. So this means they're strong. These three groups of bonding are all weak interactions. So tertiary structure, because it's primarily made up of these three groups, hydrogen bonds, hydrophobic, and then hydrophilic interactions, tertiary structure, just like secondary structure, is not that strong relative to primary structure of proteins. I guess I should also say that this is the first level with a functional protein. First level of functional protein. You're not going to find a protein that's only made up of a single alpha helix or a single beta sheet. It's going to be the tertiary structure is when we're going to see a functional protein emerge. Okay, our last level of organization is called quaternary structure. And the difference between a quaternary structure and a tertiary structure is that it's made up of multiple polypeptides. We call these multiple polypeptide chains subunits. So you can see here my not so great drawing of a quaternary structure protein and maybe a better one from the textbook. So here's our tertiary structure right here. It's composed of one long chain, and you can see maybe more clearly in mine that it's one long chain of amino acids. There's no break in the chain. And here's the same subunit, so this is one tertiary structure protein, but now it's associated with three others to make four total subunits. Now, many proteins are tertiary structure proteins, and then also many proteins are quaternary structure proteins. If you've ever heard of hemoglobin, it's the protein that fills your red blood cells. It's composed of four subunits, and it binds oxygen, helps deliver oxygen to all of your tissues. Okay, just like some of our other bonding here, quaternary structure proteins, they're all held together by hydrogen bonds. And so, again, relatively weak interactions. Okay, so over the course of the next day or two, we are going to play around with enzymes, and we're going to see what happens to enzyme function when we disrupt hydrogen bonds. That's the question we're going to be answering. What happens to enzyme function when we disrupt hydrogen bonds? And let me give you a sneak preview. It's not good. Okay, that's all.